Hello everyone. Today we are going to talk about blood and their components. So what is blood? Blood is a liquid connective tissue that fills the heart and blood vessels. The average volume of blood is 5 to 6 liters, which is about 80 kg of body weight. The components of blood are formed elements and plasma. The formed elements constitutes of RBC, WBC and platelets. Plasma contribute about 55% of the blood, RBC about 45% of the blood, WBC and platelets together form 1% of the blood. The formation of the blood cells is known as the hematopoiesis, which takes place in the bone marrow. There are three types of bone marrow: red bone marrow, yellow bone marrow, white bone marrow. Red bone marrow is the primary main site for hematopoiesis. Hematopoiesis is regulated by various hematopoietic growth factors. So, as you can see here, hematopoietic stem cells, which are the multipotent immature cells present in the bone marrow, are responsible to develop into many other mature cells. So, this HSC cells gives rise to myeloid progenitor and lymphoid progenitor. Myeloid progenitor further gives rise to megakaryotes, erythrocyte, mast cells, and myeloblasts. Megakaryotes are the thrombocyte, which is nothing but the platelets. Erythrocyte are the RBC cells. Myeloblasts give rise to monocyte, neutrophils, eosinophils, and basophils, which is nothing but the WBC cells. And this lymphoid progenitor gives rise to natural killer cells and the lymphocytic cells, which is responsible for the immunity in our body, and we'll be discussing more about it in our further videos. So, in short, the general steps for the hematopoiesis is the HSC stem cells gives rise to the progenitor cells, which gives rise to the precursor cells, which finally forms the mature cells. So there are three stages of hematopoiesis, that is the mesoblastic stage, hepatic stage, and medullary stage. Mesoblastic stage happens during the intrauterine life in the embryonic stage. It happens in the yolk sac and non-yolk sac region. Hepatic stage occurs in the during the second trimester of pregnancy. It takes place in the liver and spleen. Medullary stage, when the formation of the blood cells is in the bone marrow, it's the medullary stage. It starts from the end of third trimester of pregnancy, then continues throughout the life. So now, in clinical point of view, hematological investigations are commonly performed laboratory tests because one point is the blood is very easily available, and the next thing is be it any bodily dysfunction, it easily reflects through the alteration in blood. So blood investigations are very commonly performed in medical practices, so it's very important to have adequate knowledge in the physiology of blood. So when blood is collected in an anticoagulated tube and is allowed to settle by centrifugation, three layers can be visible. The uppermost layer is the plasma layer, and the lowermost layer is the erythrocytic layer, that is the RBC. And these both layers, the plasma and the RBC, is separated by a buffy coat, which consists of the WBC and platelets. So this buffy coat preparation is very useful in detection of abnormal cells in blood. For example, in systemic lupus erythematosus, we can see LA cells in the buffy coat. LA cells are the lupus erythematosus cells, which is nothing but the neutrophilic or macrophage that has denatured nuclear material. So that's it for today's video. Let's meet in the next video. Bye guys.